Everyone is live on YouTube now.
Okay. So good afternoon, one and all. So we are here for the World Congress 2K21 organized by Faculty of Pharmacy, Orient University, Indore. So almost we have the gathering of our eminent international speakers, and our Honorable Chancellor, Vice Chancellor, Pro-Chancellor, Chancellor sir will join very soon. We will start inauguration ceremony. So please be patient. Once again, welcome to Dr. Idris Shamat Atitala, Dean Faculty of Medical Technology, Omar Al Mukhtar University, Libya. Welcome, sir. Harika madam, make it full screen again. No, ma'am, it's not full screen now. Yes, ma'am. Now it's fine, ma'am.
Sir, Dr. Dhruv sir is here.
Okay. So we have with us Honorable Pro-Chancellor, Mr. Dr. Dhruv sir. Sir, on behalf of Faculty of Pharmacy, I welcome sir in World Congress, sir.
Thank you, Nitesh.
Welcome, sir. So just we are waiting for the chief guest, and then we will start inauguration ceremony, sir. within 2 minutes.
Dhruv sir, you are in Sanchi.
Welcome our Dean Academics, Madam.
So I was attending a program actually, and in that we were supposed to put background of our country, some place. So I am from MP. That is why I put Sanchi. That is why I put Sanchi is.
Fine, sir.
I'm changing it now.
So on behalf of Faculty of Pharmacy, I welcome Dr. Garima Ghai ma'am, our Dean Academics, Orient University, Indore. And we are also having with this Honorable Vice Chancellor, Professor Dr. Sunil K. Somani, sir. Sir, on behalf of Faculty of Pharmacy, Orient University, welcome you in World Congress, sir. Welcome, sir. Sumit, sir, kindly check our Honorable Chief Guest has joined or not.
No, sir.
Sumit, sir, please check the Dr. Sanjay, sir. Honorable Vice Chancellor, sir, has joined the World Congress. So I welcome on behalf of Faculty of Pharmacy, our Honorable Dynamic Vice Chancellor, Professor Sunil K. Somani, sir, on this platform for the inauguration ceremony of World Congress.
Yes, please. Yep, please.
Yes, Dr. Idris, we are able to listen you. Welcome, sir. Welcome in World Congress as a international keynote speaker.
Yes, please. Can I start right now?
Yeah, just sir, we are starting within 2 minutes. We are waiting for Honorable Vice Chancellor, sir. He is busy in important assignment. So we are starting the ceremony within 2 minutes, sir.
Okay, I'm ready. When you
Yes,
give
sir,
me
chalu kareenge.
signal.
Okay, Honorable Vice Chancellor, sir, is with us. So, sir, with the permission of you and Honorable Pro Chancellor, Honorable Vice Chancellor, sir, can we start World Congress, sir?
Yeah, please.
Yes,
Please start.
Thank you, sir. So I requesting host of the event, Supriya ma'am, please start the ceremony.
Sir, am I audible, sir?
Yes.
Okay. So very good afternoon to all dignitaries, guests, and delegates. With immense pleasure, I, Supriya Shidhe, on behalf of Faculty of Pharmacy, Oriental University, Indore, feel privileged to extend my warm welcome to all present here for the World Congress on Drug Discovery and Advances in Pharmaceutical Sciences, supported by International Journal of Pharmaceutical Sciences and Research. The Congress, spanning one day, includes scientific lectures by renowned international and national speakers, which gives us an insight into the latest developments in pharmaceutical drug discovery, medical, and bioscience. As we have glorious tradition of worshipping God before starting any event, so I would like to request all the dignitaries and attendees for Saraswati Vandana.
अज्ञानता से हमें तार दे माँ, शारदे माँ, शारदे माँ, अज्ञानता से हमें तार दे माँ, शारदे माँ, शारदे माँ, अज्ञानता से हमें तार दे माँ, शारदे माँ, शारदे माँ, अज्ञानता से हमें तार दे माँ.
Thank you, ma'am. Now, moving forward, I would like to request Mr. Gaurav Thakral, sir, Honorable Pro-Chancellor, Oriental University, Indore, to present a floral bouquet to the chief guest, Mr. Sanjay Singh, sir, General Manager, Glenmark Pharma, Baddi, Himachal Pradesh.
Ma'am, actually, Honorable Pro-Chancellor, sir, is busy in some important assignment. So I request Honorable Vice-Chancellor, Professor Dr. Sunil K. Somani, sir, to do the honor for our chief guests of today World Congress.
जी sir.
Yes, sir. Sir, please welcome our today chief guest of the World Congress, Mr. Sanjay Singh, General Manager, Glenmark Pharma, Baddi.
Gaurav sir.
Achha, yes, sir. Sir.
I welcome Sanjay sir.
Thank you, sir.
Thank you, sir. Once again, I would like to request Professor Dr. Sunil K. Somani, sir, Honorable Vice-Chancellor, Oriental University, Indore, to present a floral bouquet to the guest of honor, Dr. Shashi Alok, sir.
Shashi sir, namaste. Welcome to the Congress.
Next, please.
Thank you, sir. Now, I would like to request Professor Dr. Dhruv Ghai, sir, Honorable Pro-Vice-Chancellor, Oriental University, Indore, to present a floral bouquet to the speaker of today's scientific session, Dr. Idris Hamad Atitalia, sir, Dean, Faculty of Medical Technology, Umar Al-Mukhtar University, Libya.
Very warm welcome to you, sir. Dr. Idris.
Yes. Did you hear me, brother?
Yes. A warm welcome to you.
Thank you very much indeed for your kind invitation. I'm glad to be with you guys, and hope I can give you good talk about drugs discovery right now worldwide.
Sure.
My talk under the topic drug design discovery. I will give some information regarding the drug discovery worldwide right now. Many drugs with different targets are well known right now, and by natural sources and scientific sources, the discovering and the developing on the drug great to be increased. If you look at the figure diagrams here, you can see the steps for the development of the drugs. updated, and the profile of the today of our pharmaceutical business.
Dr. Idrish, we will continue a scientific session after this floral welcome and inauguration ceremony. So
Yes.
firstly, we are going for the
Looking
inauguration
forward to
ceremony.
listening to you, Dr. Idris.
Yeah.
It's like an interesting talk.
Yeah, thank you so much. Yeah, next please.
Okay,
Yes.
thank you very much, sir. Now, I would like to request Professor Dr. Garima Ghai ma'am, Dean Academics, Oriental University, Indore, to present a floral bouquet to the speaker of today's second scientific session, Dr. Hossam Bahagat, sir, Associate Professor, Medical Research Institute, Alexandria University, Egypt.
Welcome you, sir, to Oriental University.
Dr. Hossam Bahagat, sir, can you hear us? Ma'am actually sees he's here, but I think maybe, yeah.
Okay, okay.
So thank you. Next.
So anyway,
please.
yeah, we welcome him.
Yes, ma'am. Thank you, ma'am.
Thank you, ma'am. Now, I would like to request Mrs. Sonia Thakral, ma'am, Director Law, Oriental University, Indore, to present a floral bouquet to the speaker of today's scientific session, Dr. Ankur Sharma, sir, Senior Pharmacometrician, CSL Behring, US.
I think Mrs. Sonia Thakral ma'am busy somewhere else in an important assignment. So I once again request Dr. Garima Ghai ma'am to do
Yes,
the
yes.
welcome. Yes.
So I welcome Dr. Ankur Sharma
Yes, ma'am.
for
Thank
today's session. Welcome you, sir.
Thank you, ma'am. To give a red carpet welcome to everyone assembled here, now I would like to request Professor Dr. Nitish Kumar Jain, sir, convener of World Congress, Dean, Faculty of Pharmacy, Oriental University, Indore. Please, sir.
Okay. Thank you for the opportunities. So again, it's a privilege and it's an honor. So good afternoon, one and all. Welcome you all in World Congress 2021, organized by Faculty of Pharmacy, Oriental University, Indore. Oriental University, Indore is having a vision to provide a distinctly Indian environment of educational and research excellence while changing the academically accelerated individual and accelerated the academically changed individual. Continue with this motivation, the Faculty of Pharmacy, Oriental University, Indore is going to organize World Congress on Drug Discovery and Advances in Pharmaceutical Sciences, supported by International Journal of Pharmaceutical Sciences and Research. World Congress 2K21 is based on the theme Drug Discovery and Advances in Pharmaceutical Sciences, is conducted by the Faculty of Pharmacy. The conference, spanning a one-day, that is 10th December 2K21, includes various scientific sessions by the renowned international speakers from Egypt, Libya, and US. They will share the latest and insights into the latest developments in the pharmaceutical drug discovery, medical and biological sciences. More delegates are expected to participate from all over the globe. The unique event explored the significance of new trends, ideas, research experience, foster collaborations across industry, academia, and evaluate emerging pharmaceutical technology across the globe. The World Congress is aimed to provide a unique platform for the professionals, researchers, academicians, industry delegate, and allied field of pharmaceutical science to interact and share their experience and knowledge on recent development on drug discovery and recent advances in the biological sciences. The World Congress is organized under the valuable guidance and blessings of Honorable Chancellor Sri Praveen Thakral sir, Honorable Pro-Chancellor Engineer Gaurav Thakral sir, Honorable Vice Chancellor Professor Dr. Sunil K. Somani sir, Honorable Pro-Vice Chancellor Professor Dr. Dhruv Ghai sir, Honorable Dean Academics Dr. Garima Ghai ma'am, Director Law Mrs. Sonia Thakral ma'am, our Registrar Dr. Pradum Yadav sir, and entire fraternity of the Oriental University Indore, and on behalf of Faculty of Pharmacy, Oriental University Indore, I welcome all the keynote speakers, Professor Dr. Idris Hamad Atitala from Libya, Dr. Hisham Bhagat from Alexandria University, Egypt, and Dr. Ankush Sharma from Bahrain, US. And on behalf of Faculty of Pharmacy, Oriental University, Indore, I welcome Honorable Chancellor, Sri Praveen Thakral, sir, Honorable Pro-Chancellor, Mr. Engineer Gaurav Thakral, sir, Honorable Vice Chancellor, Professor Dr. Sumani, sir, Honorable Pro-Vice Chancellor, Dr. Dhruv Ghai, sir, Dean Academics, Professor Dr. Garima Ghai, ma'am, and Director Law, Mrs. Sonia Thakral, ma'am, Principal UIP, and all my dear senior doctorate faculty, student and PhD, PG research scholar, and all the delegates and judges of the scientific session. So on behalf of entire fraternity of Oriental University Indore, I welcome you all in World Congress 2K21. Thank you. Thank you so much.
Thank you, sir, for giving us insights about the World Congress. Now, I would like to request Honorable Professor Dr. Dhruv Ghai, sir, Pro-Vice Chancellor, Oriental University, Indore, for inaugural speech. Please, sir.
Yes, thank you, Mrs. Supriya Shidhai. Very good afternoon to all. Honorable Vice Chancellor, Professor Dr. Sunil Somani, other deans of the university, keynote speakers, delegates, and my colleagues, ladies and gentlemen. With enormous pleasure, I extend a very warm welcome on behalf of Oriental University Indore, which has convened this World Congress on Drug Discovery and Advances in Pharmaceutical Sciences through virtual mode. The objective of this conference is to utilize the knowledge and experience of various innovative researchers and to share them with others to maintain the productivity of discovery and development of drugs. Pharmacy profession is committed to providing a transformative learning experience in collaborative and diverse environment, focus on improving the healthcare system to serve the community in the development of new drugs. I wish to convey heartfelt thanks to all the participants for their presence, and also would like to express my sincere gratitude to the guests and keynote speakers from various countries who have readily agreed to spare their valuable time to enhance our knowledge. We are indeed elated and obliged to have such venerable and illustrious people as our guests. At this point, I would like to extend a warm welcome to our guests, Mr. Sanjay Singh, General Manager, Glenmark Pharma, Baddi, Himachal Pradesh. Dr. Shahi Alok, Editor-in-Chief, IJPSR. I would also like to thank them for accepting the invitation for being present at this occasion. I would also like to extend a warm welcome to our keynote speakers, Dr. Idris Hamad Atitala, Dean, Faculty of Medical Technology, Omar Al Mukhtar University, Libya. Dr. Hosam Bhagat, Associate Professor, Medical Research Institute, Alexandria University, Egypt. So you guys are operating on very different time zones. So we thank you for making time for us. Dr. Ankush Sharma, Senior Pharmacometrician, CSL Behring, USA. Also, I would like to congratulate the convener, Dr. Nitesh Jain, and the entire pharmacy department, both the schools, UIP and University Institute of Pharmacy and Oriental College of Pharmacy and Research. Organizing Secretary, members, Dr. Sumit Dwivedi, Dr. Niharika Gokhale, and all the other members of the organizing committee. And pleased to declare this World Congress open. Best wishes. Thank you.
Thank you, sir.
Thank you, sir, for your kind words. Now, I would like to request Professor Dr. Sunil Somani, sir, Honorable Vice Chancellor, Oriental University, to please bless us and bless the gathering also. So please.
Good afternoon, everybody.
Good afternoon, sir.
Yeah. I welcome every member who is present for this World Congress on Drug Discovery and Advances in Pharmaceutical Sciences. It's a great attempt by the Faculty of Pharmacy of Oriental University to organize not only this World Congress, but at the same time able to attract the people from all over the world. And I am thankful to all of them for joining for this World Congress. See, when we recall the days when March 20 was there, and particularly for India, however, it was December 19 when the COVID virus first attacked this particular world, and gradually it is spread all over, and then we realized in March 20. Why I'm mentioning it? Because here comes the very noble profession of pharmacy comes into the picture. And whatever drug discovery, we have seen that since inception of the pandemic till we get the different vaccination by different companies from different parts of the world also. This industry basically worked a lot in getting all those vaccination doses and for that sake some other medicines as well, so that the people can be cured or at least get relieved for and those hectic days. Nobody, particularly this generation who has faced that trauma, cannot forget, and at the same time they will not forget the whatever advances in the pharmaceutical sciences and drug delivery was there between those, say, 8 months or 9 months. It was tremendous effort by the community, which we call as the pharmacists. This particular World Congress is dedicated on the drug discovery as well as the advances in this area. All the learned speakers who will share their knowledge will definitely be very helpful to all the people who are attending this. And it is really very important that the different people will take advantage of their knowledge, and ultimately our effort for organizing the World Congress will come forward. I give all the best to all the participants who are there and congratulate the organizers for such events. And from on behalf of, say, Oriental University, I am thankful to everyone. Namaste.
Thank you so much, Honorable Vice Chancellor, sir, for your valuable blessings and your kind motivation. Means a lot to us, and definitely hope this World Congress will take a good lead, and we will give recognition of Faculty of Pharmacy at global level. Thank you so much, sir.
My pleasure.
Thank you, sir. Now, I would like to request all the dignitaries to please join us in souvenir release.
Yes, please start, ma'am.
Yeah, great.
Sir, almost we have received 115 abstracts. So we are publishing this proceedings with International Journal of Pharmaceutical Science and Research, which is index in the product of Science Direct. So this is something but we are going to plan, and it will become as conference proceedings.
Right, very nice. Very
Thank you,
nice.
sir. Yes, next, please.
Thank you, ma'am. Thank you, sir. Now, coming to the end of inaugural ceremony, I would like to request Professor Dr. Sumit Dwivedi, sir, Organizing Secretary, Principal, University Institute of Pharmacy, Oriental University, Indore, to extend vote of thanks.
Thank you, Supriya Madam. So it is indeed a very great moment for me to express the vote of thanks for today's event of inauguration session, that is the World Congress on Drug Discovery and Advancement in Pharmaceutical Sciences. I take this opportunity to thank our Honorable Chancellor, Shri Praveen Thakral sir, Honorable Pro Chancellor, Shri Gaurav Thakral sir, Honorable Vice Chancellor, Professor Dr. Sunil K. Somani sir, Honorable Pro Vice Chancellor sir, Dr. Dhruv Ghai sir, respected Dean Academics, Dr. Garima Ghai ma'am, and entire management that they have given us opportunity to conduct this particular event. At this particular point, hello. At this particular point, I take opportunity to thank our chief guest, Mr. Sanjay Singh sir, General Manager, our guest of honor, Dr. Shashi Alok sir, Editor-in-Chief, IJPSR, a group of Science Direct Journal, that they have accepted our invitation to be a part of this World Congress. I am also thankful to all our speaker, Dr. Idris Ahmed Atitala, Dean, Faculty of Medical Technology, Libya, Dr. Osham Bhagat, Associate Professor, Alexandrian University, Egypt, and Dr. Ankush Sharma, Senior Pharmacometrician, US. That in a very short duration, they all have accepted our invitation and they are a part of this World Congress. Thanks to Dr. Shatendra K. Srivastav sir, that they judges the poster presentations and given their valuable insight. Last but not the least, I am thankful to each faculty for their contribution for making this World Congress a grand success. Thank you.
Thank you, Dr. Sumit Dwivedi, for vote of thanks. As we know all that our honorable dignitaries are very busy with some important assignments. So we are moving further for scientific sessions, sir. So if you can stay with us, that will be an honor to us. And thank you so much to all the honorable dignitaries, Honorable Vice Chancellor, sir, Honorable Pro Vice Chancellor, sir, Dean Academics, madam, for sharing your views and giving your valuable time. So now we are moving further for the scientific session of this World Congress. Thank you so much, sir. Thank you.
Thank you, Jinsab, and go ahead, continue.
Thank you, sir. Thank you so much, sir.
Sir, we are here with the video.
Okay. Yes, ma'am, please start. Requesting Supriya ma'am, please start the scientific session. So requesting Shukriya ma'am, please hand over the things to a scientific community so that we can start a scientific session of international keynote speakers.
Now we'll move forward towards the scientific session.
तो मैडम आपका सबका रेडी में
For the same, I request Gulfisha Sheikh to please take over the mic. Over to you, ma'am.
Requesting faculty, please mute yourself. Deepak sir.
Thank you, Supriya ma'am. I, Gulfisha Sheikh, wish you a very good afternoon to all. With a great joy, I feel privileged to extend my warm welcome to our today's speaker for first scientific session, Dr. Idris Hamad Atidullah, sir. It's a great pleasure for me to introduce our speaker, Professor Department of Botany, Faculty of Science and Department of Microbiology, and Dean of Faculty of Medicinal Technology, Omar Al Mukhtar University, Al Baida, Libya. Professor Atidullah completed his PhD from Uppsala University, Sweden, and postdoctoral studies from USM University since Malaysia. He is the Director of Research and Study at Agriculture Research Center and Scientific Consultant, Libya. Sir has published more than 120 papers in reputed journals and serving as managing editor of International Journal of Pharmacy and Life Sciences and editorial board member of more than 65 journals. Sir has written dozens of books, among them few are in Arabic language about algae, translated book of proteomic and edited two books to be published by Taylor and Francis, USA. He has done several international collaboration with academics and industry. In India, he has worked with Dr. Sumit Dwivedi, Principal UIP, Oriental University, Indore, MP. Not but the least, his major research interest lies on microbiology, molecular biology, bioscience, mycology, and molecular evolution. Now, I request to Dr. Idris Hamad, sir, to take the audience in his hands. Welcome you, sir.
Sir, please unmute. Idris, sir, please.
Make a co-host also so that he can share his slides. Yeah, Dr. Idris. Hello.
Idris, sir, are you there?
Yeah, Dr. Idris, is there any technical issues from your side?
Right now, I start. Did you hear me right now?
Yes, yes, please. You can share your
Yes.
presentation, sir.
Yes. Thank you very much. Thank you very much for your invitation. I'm introducing myself as Professor in microbiology and molecular evolution.
Okay.
I express my gratitude to Professor Somit, who really give me the confidence to be with you guys.
Thank
Hope
you.
my lecture can give some knowledge and increase knowledge in the topic of drug design and discovery. This topic right now worldwide uh, encourage people to get drugs with high impact to stop different diseases which comes from different hosts and different organisms. Right now, the discovering and the development of the drugs has been increased, and that uh, due to many different competition between companies, and the target for that today it about time, how time need to get good drugs. By contrast, the chemists developed new device in the 3 months, but to need to why? Because the biochemical animal and human trials color approval to be in the end of this discovering the drug. Medicine is very important, and medicinal plants it really encourage people to get good drugs. Medicinal chemists today are facing serious challenge because of the increased cost and enormous amount of time taken to discover new drugs, and also because fierce competition among different drugs companies. That's why I come back to the competition. And if you look at the new the next slide, the drug design showed to be from uh, uh, the start point and to the end. And when we get good chemical and doing that tries in the target animal, we need several steps to get different scales and different bioassaying methods to get finally a good drug in the end. That lead to to develop new markers, especially molecular markers. And by then, we get to have good animal, but that animal has in the end with impact drugs, and the drugs can be used in term of human. And to move that from the target trials animal after different assay to the human, we need many different experimentations, and that really hard for people working in the pharmacy and medicine. The history of drug discovery since 1909, first orientation drug design, the goal safer syphilis treatment than atoxide. After that, we can talk about this different scenarios, and that wanted to maximize toxicity to pathogen and minimize toxicity to human. And this need really different groups from different departments to work together to get good design for good drug impact. That they found telemedicine, which has replaced the penicillin in 1940, and that design of that drug, which moved the worldwide to the revolutions of medicine. 1960, first successful attempt to relate chemical structure to biological and quantitatively quantitatively structured activity relationship, and that people don't know about many different ways has been done to have that penicillin in experimentation in different countries from the beginning. Mid to late 20th century, understanding to understand disease status, biological structure, process, drug transport, distribution, and metabolism. What I'm talking about right now, we need different biochemical activities done before we can test the drug for human. Medicinal chemists use this knowledge to modify chemical structure to influence drug activity stability. And we look for the stable compound which can be used for as a treatment or as a drug. The broxine for local anesthesia, for example, and broxidine and anthramic. Those compound which has been developed made different biological and biochemical and experimental by chemists before they do test in the animal and move it to human. Drug discovery overview, we are talk about approaches to drug discovery. For example, when we talk about sustainability, like chemist modification, screening, and additional need different screening before we can develop good compound and structure. Serendipity, the chance favor the prepared mind. In 1928, Fleming studied stuff, but contamination of blades with airborne mold fungus noticed a bacteria were delayed in the area of the mouth. A mold product inhibited the growth of the bacteria, and that as antibiotic penicillin. And when we look at the start from the Fleming, Alexander Fleming, when he developed the revolution of penicillin from the beginning, which saved many human population to be survived. Chemical modification can be go through many different steps. A homolog approach, homologs of lead prepared, and molecular disconnections, simplification, and molecular addition, and isoelectric replacement. And when I talk about those steps, and we go to the chemical modification, and we need good chemists and good pharmacists, which can be work together to get good design for the drug. When they talk about traditional method, we know from the beginning, a analog of non-active compounds synthesized with minor modifications that will lead to improved biological activities. And when you see that compound in the slide, you can see how big job done to have good staple compound to be used as a drug. Advantage and limitation end up with something with very similar to what you start with. It means you need to go to second step of screening, testing random a large number of different molecules of the biological activity reliably. And innovations have led to automation, synthesized computational synthesis, and testing high throughput screening. Example, when you talk about Bronzesteel is derived from dye that exhibited antibacterial properties, and we need to see what concentration, how many colony forming units can can be decreased when the drug has been tested. A rational based of serendipity, intuition, trial and error approach, time consuming with low through output, no design mostly me to approach. Rational drug design, when you talk about simildine segment, uh, start with validate biological target and end up with a drug that optimally interact with target and trigger this biological action. Then we work with one step or two step from the reactions. And when you talk about problem, histamine trigger release of stomach acid want to a histamine antagonist to prevent stomach and released by histamine validated biological target. Histamine can trigger many signaling in the stomach with interacting together. Histamine analog very synthesized with systematically very structured chemical modification and the screening in guanyl histamine showed some antagonistic properties and lead compound. Relation drug design settlement segment continued. When you talk about different steps, when you talk about chemical modification, well made of the lead optimization, and then we go to more potent and orally active when you get orally. But Urea found to be toxic in clinical trials when we tested. And we go to replacement of a group lead or an effective and well tolerated product. We need to modify it and then we let reaction to be stable. Eventually replace the Azenta with improved safety profile. When you get to safe reaction. First generation rational approach in the drug design. In 1970s, the medicinal chemist considered molecule as a topological entity in two dimensions with its associated chemical properties. OSAR concept became quite popular. It was implemented in computer and constituted first generation rational approach to drug design. When we get to make a good design using computer software. Second generation rational drug design, the acceptance by medicine and chemists of molecular modeling was favored by fact that also was now supplemented by three visualization, the lock and the key competent, actually supported by three model, computer aided molecular design is expected to contribute to intelligent leads. When you go to this slide, we talk about the step as usually we used to have most rational approach as I talked earlier. Revolutionary drug designing, which is a very important part in my talk. Instant time, natural product with biological activities used as a drug. We are focusing on using natural product to be used under taking under consideration any toxic side effects. And to avoid this toxicity, we need a evolutionary approach. And the chemist era, synthetic organic compound, and the rationalized design process, SARS, and computational chemistry-based drugs. And then I go back to the point that drug need chemists with computational guys to work together to get all steps under consideration. all are wearing under consideration biochemical error to elicitate biochemical pathway and macromolecular structure as a target as well as a drug. Drug discovery process. We have different points I can minimize and can summarize in this scheme. Target identification and validation. And we need chemical libraries, computer chemist, natural product, high throughput screening, and we will work together in this part to lead compound, evaluation compound, and then we can go to human trials on clinical trials. New target, total genome. We have then genome people. They know genes. They know the risk of using molecular process without knowledge and taking together. Then we go to big discovery. Anyway, for vaccine, for whatever we will look for, to stop disease to be aggressive, to stop high virulence factors to be stop, need to work with different approach. We know from our experience two types of organism or whatever organism attack people whatsoever parasite, virus, bacteria, fungus, whatever, we need to take under consideration the variance factor. We know that high variance factor and low variance factors, and this is related to the evolutionary point of from evolutionary point of view to get those together can look can between. This is my evolution and the total genome from any governance, then we can see from this figure and including biological space, small molecular space, and new drug space, and excising the drug, how many targets we need to get the genome under consideration. High throughput screening, functional integration, biology, chemistry, screening, technology, and information. These many different factors, multifactorial need to be put together when you are interested to work on drugs. Smart drug discovery platform, when you talked about gene target, therapeutic target, a new ligand, chemical candidate, and by what we say biochemical or by throughout medicine, and we have the biology and chemistry working in different parts. Functional genome, structural genomic, proteomic, and bioinformatic in one part, and then in vivo disease model, admit and drug delivery, and principal toxicology, those in the second part. And in between novel ligand, when you have structure, synthetic high throughput of biology screening, and in the end, in vitro biological mechanism, and we can understand the mechanism of that target to in term of gene or in term of chemistry. A view drug discovery roadmap is illustrating some key multidisciplinary technology that enable to development of breakout robot medicine from the promising candidate and generation process that are related to novel ligand. And this is from Nature in 2006, but has been published. Molecular modeling, when you have this scheme, is very simple but very hard to understand. And when you talk about NMR technology and X-ray technology, structure dissemination And when it talks in second part, and when I have structure drug design relation, drug design, and we get to the output of bioinformatics, chemoinformatics, and the competition of chemistry underway. When you have those technology working together in the homology modeling comes from this mixture, you would see model constructions. When you construct the compound, molecular mechanism, competition, and also molecular biomarker. When you see the next slide, when you see in how steps in the figure, stage by stage quality assessment to reduce costly late stage attrition. How many steps you need to get this done in the end of the experimentation. The drug discovery overview, lead discovery, identification of compound, and trigger specific biological action. The lead optimization properties of the lead and tested with biological assay. New molecule and these are designed and synthesized to obtain these properties. To understand that, the kinetic modeling, what we call it extras, that starts from different parts, and the most important pharmacokinetic would take 56% from the total, and the toxicity she takes 16% from this figure, and that's why pharmaceutical people, chemists, should go to toxicity bar and work with that bar to get drugs with very low toxicity. And the drug design right now with high risk of toxicity, they use it to competitions of different companies, which the protect with safety and ethical in working together to get the drugs. It's really important for people work in this subject. Changing criteria for the clinical candidate. We need properties activities. When you get to high or good ligand or good drugs, you need to be working in those topics together. Drug discovery is judging act dynamic process of co-optimization. From the schedule, we have the activity and the save, as I mentioned earlier. And the save, when you say to the next slide, the profile from metabolic stability, solubility, etc. When you get a solution stability, and to get this cytotoxicity, those together lead to drug must survive in vivo pairs from dose to target, from stomach to cells. Yes, indeed. When you see the stomach active in the drug active in the stomach, and the disease has been decreased, you should go partly from intestine to blood to organ to cells and see cell-mediated organism how immunity give that given by that drug by that drug to the target, and study cell biology after those assay is indeed it. And each stage of biology has properties issues from HTS, enzyme assay, cellular assay, animal to human. And that is very tricky when you get part from part. You need to study and to know that this part well before going to the next part. Identify purity, solubility,
Idris
stability,
sir,
and pilot
Idris
state.
sir, hello.
Yes, yes, sir.
Sir, time is going to end, so can you conclude within a minute?
Yes, I'm going to the last slide, please. Okay.
Okay, okay, sir. Okay, fine. Thank you.
Give me just a few minutes because I want to summarize my talk. When you talk to, because this slide is very important, sir. When you talk from the start to the animal to human, the main part, stability, plasma stability, instability, solubility, and those different factors need us to get to the drug discovery is multidisciplinary, complex, costly, and intellect intensive process. Modern drug design technique can make drug discovery process more fruitful and rational, but knowledge management technical specific expertise can survive time and cost, which is paramount need to be to the hours. What I'm going to say, governments, companies must attractive, good expertise, good faculties, has good history in this topic to get high impact drugs. Otherwise, it will go to the risk because we will when we produce drug without taking those several factors together, the drugs can be successfully for a while, but fail for a long destination. And this is from my knowledge point of view. When those viruses coming recently, Corona, for example, the companies competition lead to produce good vaccination, but nobody knows how much cost those vaccination paid by that life, by human life. And now many different problems coming up. Yes, it decreased the disease, but because the study was very short, I'm I'm expecting failed to this vaccination to be working really active for long time, not not for to say this vaccination is not good, but to say we have to attract people worldwide, taking environment, evolution, societies under consideration to lead to the end to give world our world and globally a good vaccination. And this is our hope. Thank you very much indeed for listening. I'm sorry for taking time. Thank you, Professor Sumit, and your colleagues for your confidence, invited me. Hope I gave something good for you and for the audience. Thanks for your university and your president of vice president of university, and wish you good luck.
Thank you so much, sir, for your valuable present you have given about the insight of drug development and discovery. Thank you so much.
Thank you, really. Thank you, Dr. Idris. You have very good scientific knowledge about the toxicity, especially. So I appreciate the concept which you have shown us with the participants of the World Congress. And thank you so much for giving your valuable time as an international keynote speaker. Thank you so much.
Thank you.
Thank you, sir. Now, I would like to request Professor Dr. Nitish Kumar Jain, sir, Dean, Faculty of Pharmacy, Oriental University, Indore, to present token of remembrance to our today's speaker, Dr. Idris Hamad Atitalia, sir. So, please.
Okay, so thank you once again. Thank you so much. So, sir, on behalf of Faculty of Pharmacy, Oriental University, Indore, please accept this token of appreciation as being a international keynote speaker. So, I am presenting this appreciation as a token of appreciation to Professor Dr. Idris Ahmad Atitala, Dean Faculty of Medical Technology, Omar Al Mukhtar University, Libya. Thank you so much, sir, for your valuable presence and grace the occasion as an international keynote speaker. Thank you, sir.
Thank you, sir. Moving towards the next scientific session, I request Ms. Deepika Bairagi to please take over the mic. Over to you, ma'am.
A very warm good afternoon to all of you. Thank you so much, ma'am. I hope you all have enjoyed the session of Professor Dr. Idris Hamid Atitullah. Now, on behalf of entire fraternity of Oriental University Indore, I, Deepika Baragi, Assistant Professor, Oriental University Indore, welcome our next scientific session keynote speaker, Dr. Osam Bhagat Alkanfani. He is an associate professor at Medical Research Institute, Alexandria University, Egypt, and also the head of Public Health Department for Omar Al Mukhtar University. Dr. Osam Bhagat communicates clinical biochemistry, immunology, laboratory medicine to laboratory medicine students and public health students. His research interest is in clinical chemistry and laboratory medicine. He has more than 20 research as well as review paper in various international and national journals. He has been assigned as editorial board member of several international journals. Dr. Bhagat has wide knowledge of drug development as his keen interest is in drug discovery and development for type 1 and type 2 diabetes. There is a lot to say about Dr. Bhagat, but due to lack of time, I invite Dr. Bhagat to start his session. Over to you, sir.
Yes, requesting Dr. Husham Bhagat, sir, please share your presentation as international.
Bhagat sir, you are mute. Please unmute yourself.
You hear me, sir? Sir, you hear me?
Yes,
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 yes.
sir.
Okay, good afternoon for everyone.
Good afternoon, sir.
Afternoon. I am Hussam Bahgat, clinical associate professor of clinical biochemistry in Omar Al Mukhtar University. I appreciate your invitation to speak about my lecture. My topic about insulin therapy in adults with type 1 diabetes mellitus. First of all, we all know about type 1 diabetes mellitus is a result of the destruction of insulin producing pancreatic beta cells, leading to insulin deficiency. The destruction of beta cells is usually the result of an autoimmune process, but in rare cases, the cause may be unknown. Type 1 diabetes mellitus was originally believed to develop mainly during childhood or adolescence, but it is now known that it can develop over the whole life span, even in individuals aged over than 80 years. Most patients with type 1 diabetes mellitus are adults, partly because children diagnosed with type 1 diabetes mellitus will survive to adulthood, but also because of new cases diagnosed in adults. The identification of type 1 diabetes mellitus may be challenging, and the type 1 diabetes mellitus may be misdiagnosed as type 2 diabetes mellitus in adults aged over than 30 years. Latent autoimmune diabetes in adults, a form of type 1 diabetes mellitus that involves a slowly progression immune mediated loss of beta cells, may comprise 5 to 15% of type 2 diabetes mellitus cases. Proper identification of type 1 diabetes mellitus is extremely important because these patients need to receive immediate insulin therapy and reach optimal glycemic control to prevent the onset of diabetic ketoacidosis, which is associated with an increased risk of death, and to delay microvascular and macrovascular complications. Patients with type 1 diabetes mellitus require lifelong insulin replacement therapy from the time of diagnosis. Many insulin options for type 1 diabetes mellitus treatment are currently available, including human insulin and insulin Analogues, the American Diabetes Association, and the UK National Institute for Health and Care Excellence guidelines recommended against routine testing of C-peptide or autoantibodies for diagnostic purposes. But this could be used for clinical questionable cases on the basis of the natural history of type 1 diabetes mellitus. Three stages of type 1 diabetes mellitus development were proposed. Stages 1 and 2 are both pre-symptomatic with detectable beta cells autoimmunity, with stage 2 additionally including dysglycemia, impaired fasting glucose or impaired glucose tolerance. And the stage 3 is symptomatic with new onset hyperglycemia. Glycemic targets. The large scale prospective diabetes control and complications trial conclusively showed that intensive glycemic control with basal bolus insulin treatment regimen reaching glycated hemoglobin HbA1c levels of approximately 7% is associated with reduced microvascular risk with respect to retinopathy and neuropathy and diabetic kidney diseases, and reduced long-term cardiovascular risk compared with conventional glycemic control with fixed dosing insulin regimen, reaching HbA1c level of approximately 9%. This results from the basis of current guideline recommendations in general evidence-based glycemic target recommendations by the ADA, Diabetes Canada, and NICE for adults with type 1 diabetes mellitus are all in agreement. All recommended an HbA1c goals of less than 7% for most non-pregnant adults, or less than 8% for patients with a history of severe hypoglycemia or severe complications or comorbidities. This is level B. And less than 6.5% if this goal is achievable without the risk of significant hypoglycemia or other adverse events. This is level C. Similarly, Diabetes Canada recommends a target HbA1c of less than 7% for most patients with type 1 diabetes mellitus. NICE recommends a target HbA1c of less than 6.5% for most patients with type 1 diabetes mellitus, but suggests considering a person's daily activities, aspiration, likelihood of complications, comorbidities, occupation, and history of hypoglycemia when setting individualized targets. This table, I don't know, you see these tables or what? Okay, according to the ADA recommendations to achieve an HbA1c less than 7%, preprandial capillary plasma glucose should be from 4.5 to 7.5 millimole per liter, and peak postprandial capillary plasma glucose should be less than 10 millimole per liter. This equal 180 milligram per deciliter. Sorry. Similar levels are recommended by Diabetes Canada and NICE with the advent of continuous glucose monitoring systems, CGM. The time in blood glucose concentration range of 3.9 to 10 millimole per liter. This equal 70 to 180 milligram per deciliter may become an additional indicator of overall glycemic control, which emphasizes the importance of decreasing blood glucose variability in addition to mean glucose values with time in hypoglycemia, a safety marker of insulin treatment in person with type 1 diabetes mellitus. Insulin therapy. Insulin replacement is necessary for all people with type 1 diabetes mellitus and involves multiple daily injections, short-term MDI, of basal and prandial insulin, or continuous subcutaneous insulin infusion, short-term CSII level A, a variety of insulin, basal and prandial, and the modes of administration, syringe or pen or pre-filled pen and pump are available, achieving optimal glycemic control while avoiding hypoglycemia and other adverse effects, such as excessive weight gain and lipodystrophy, requires individualized insulin therapy supplemented by glucose monitoring, either intermittent self-monitoring of blood glucose with a glucose meter or CGM, and proper therapeutic education, carbohydrate counting, and insulin dose adjustment according to carbohydrate intake, activity levels, and the blood glucose values, and the proper injection technique. Basal insulin, which include long-acting insulin analogues, first generation determined, brand name Levemire, and Glargian, brand name Lantus, 100 unit per milli. Short term for Glargian, 100. Second generation Glargian, 300 unit per mil. And Digludix, or human intermediate acting insulin, neutral protamine hagedron, NPH insulin, are given 1 to 3 times a day according to their pharmacokinetics properties to control glucose levels in the fasting state and between meals. Prandial insulin include rapid-acting insulin analogues like lispro-aspart glucine and fast-acting aspart or short-acting regular human insulin and are given before each meal. And at each time, a correction of a high blood glucose value is required. Typically, prandial insulin are injected 3 times daily before main meals and are injected more often if 3 meals per day are consumed or correction pulses are required. Pre-mixed insulin, either human or analog insulins, are available but are not appropriate for patients with type 1 diabetes mellitus because the independent adjustment of basal and prandial insulin doses is not possible. Rapid-acting inhaled insulin is available for use within a limited dose range, but large trials are needed for confirmation of efficacy. Furthermore, inhaled insulin is contraindicated in patients with chronic lung disease, is not recommended for those who smoke, costs at least twice that of other rapid-acting insulins, and may require additional dosing after meals. Basal bolus insulin regimens that mimic physiologic insulin secretion, usually MDI or less frequently CSII, should be initiated from the time of diagnosis of type 1 diabetes mellitus. The starting total daily insulin dose is typically weight-based, ranging from 0.4 to 1 unit per kilogram body weight, as recommended by various guidelines. However, In some cases, particularly in newly diagnosed patients, the dose of insulin required may be less as a result of transient remission that may occur early after the initiation of insulin substitution. To prevent the risk of early hypoglycemia, a lower initial dose of between 0.3 and 0.4 units per kilogram body weight may be used. In contrast, a higher insulin dose of 0.7 units per kilogram body weight may be appropriate in the presence of ketones. In general, approximately half, 40 to 60% of the total daily dose is given as a basal insulin, and the receipt is divided into meal-related doses based on carbohydrate content. The exact insulin doses are reached by continuous titration. Prandial doses are mainly conditioned on carbohydrate intake and to a lesser extent on protein and fat content, while basal insulin is dependent on body weight and insulin sensitivity. To assess short-term glycemic control, it is recommended that most patients perform self-monitoring of blood glucose at various times, before meals and snacks, at bedtime, occasionally overnight, 2 hours postprandially, to titrate dose of prandial insulin prior to exercise when low blood glucose is suspected, after treating low blood glucose until achieving normal glycemia, and before critical tasks. CGM along with intensive insulin regimen can improve glycated or HbA1c in adults with type 1 diabetes mellitus who are not meeting glycemic targets, level A. CGM can also be useful for those with hypoglycemia, unawareness, and or those with frequent hypoglycemic episodes, level B. For maximal benefit, CGM should be used daily or as frequently as possible to reach optimal glycemic effect, level A. Pharmacokinetics, pharmacodynamics, clinical efficacy, and safety with basal insulin analogues. The basal insulin analogues have much improved efficacy and the safety profiles and increased patient satisfaction over NPH insulin in patients with type 1 diabetes mellitus. In general, use of long-acting basal insulin analogues results in fewer hypoglycemic episodes compared with the use of NPH insulin. A meta-analysis of trials testing first-generation long-acting insulin analogues versus NPH insulin found a reduction in the risk of nocturnal hypoglycemic episodes and the HbA1c level of patients using the former. Another meta-analysis confirmed these results and also indicated less weight gain in patients on first-generation long-acting insulin analogs versus those on NPH insulin. Glargine 100 versus NPH insulin is a long-acting insulin analog, which is a solution in the insulin vial that precipitates at physiologic pH in the subcutaneous tissue. This precipitates and the more stable hexamers result in a slower dissociation into dimers and monomers, leading to protracted absorption into the circulation. Glargine has a reduced peak concentration per action profile compared to NPH insulin, and a lower inter-individual variability than NPH insulin. In general, Glargine 100 once daily is comparable to or better than MBH insulin once or twice daily in maintaining glycemic control as measured by HbA1c. Similarly, decreases in fasting plasma blood glucose are comparable or significantly greater with Glargine 100 than those with MBH insulin.
Okay.
One slide.
Okay. Yes. So you are going to conclude the session, right, sir? Great, great.
Yeah, yeah.
Great
Okay.
research, sir. I really appreciate your research. You have done a lot of work in type 1 and type 2 diabetes. Yes, please, sir, continue.
Thank you very much for you, sir. Okay. Decreased weight gain in patients on Glargine 100 compared with weight gain in those on MBH insulin has been observed. Conflicting results have been found regarding hypoglycemia, with some studies reporting no difference between Glargine 100 and MBH insulin, but most reporting significantly fewer hypoglycemic events, including nocturnal events with Glargine 100 than with MBH insulin. Okay. أكتفي بهذا القدر. Thank you for all. I appreciate your invitation, sir, and I hope inshallah يعني the greatest for your session. Thank you so much.
Thank you so much, Dr. Sham Bhagat, sir. You have given a lot of scientific insights about the type 1 and type 2 diabetes. It's a very prevalence in India also, definitely. So all the researchers will get the benefit of your scientific thought about the type 1 and type 2 diabetes. Yes, continue, please.
Thank you, sir. Yeah.
Thank you, sir. Now, I would like to request Professor Dr. Sumit Dwivedi, sir, Principal, University Institute of Pharmacy, Oriental University, Indore, to present token of remembrance to our speaker, Dr. Hosam Behgat, sir, Associate Professor, Medical Research Institute, Alexandria University, Egypt. Please, sir.
Okay, so on the behalf of Faculty of Pharmacy, sir, please accept our memento for your keynote address that you have given on type 1 and type 2 diabetes. Indeed, that was a great research, sir. Thank you so much for accepting our invitation for this World Congress. Thank you, sir.
Thank you, sir. I appreciate your invitation.
Thank you, sir. Thank you, sir.
Thank you. Thank you so much.
So I think now we are going for the valedictory ceremony of this World Congress. So I request host of the event.
Yes, sir. Thank you, sir. Now, with the permission of convener and organizing secretary of World Congress, I would like to start valedictory function of this Congress. Knowledge and skills are the crops that are nurtured, groomed, and given all possible opportunities to grow and bloom. Now, the much awaited moment of the day is here, as it gives the returns to the efforts put in by the participants of poster presentation. I would like to announce the excellent poster presenters of today's Congress. First, I would request Niharika. Yes, ma'am. Ms. Tanu Bhargava. Congratulations, Ms. Tanu.
Yeah, many congratulations to Ms. Tanu Bhargava.
Thank you.
Presented paper in World Congress. So really appreciate your research. You have done a great job. So that's why you are nominated for the Young Scientist Award, right?
Thank you so much, sir.
Congratulations, Ms. Tanu Bhargava.
Thank you so much,
Dr.
sir.
Nitesh Kumar Jain and Dr. Sumit Dwivedi, sir, to please present all the certificates to the excellent participants of today's Congress. Next, Ms. Megha Joshi.
Congratulations, Megha, for this
Thank-
Young Scientist Award in World
Thank you,
Congress.
sir. Thank you very much, sir.
Many congratulations, Ms. Megha Joshi, for the Young Scientist Research Award, and wish you all the best for the future research and endeavors. Thank you.
Thank you, sir.
Ms.
Thank you very much.
Tejasri Verma.
Congratulations,
many congratulations to Miss Pooja Sri Verma
Thank you so much.
for Young Researcher Scientist Award.
Thank you, sir. Thank you. Sir.
Congratulations.
Miss Preeti Mule.
Yes. Many congratulations, Miss Preeti Mule, madam.
Thank you, sir. Thank you.
Great research, and you
Congratulations,
deserve
ma'am.
for
Thank
the
you,
Young
ma'am.
Scientist Award. Thank you.
Thank you, sir. Thank you so much. Thank you, ma'am.
Miss Radhika Kotami.
Congratulations, Miss Radhika Kotame, for getting Young Research Scientist Award 2K21 in World Congress organized by Faculty of Pharmacy, Orient University, Indore.
Miss Leena Devre.
Thank you, ma'am. Thank you so much.
Congratulations, Miss Leena Devre, for getting Young Research Scientist Award. Many congratulations. Continue with the good research and motivate the other researchers by doing this kind of research. Thank you.
Thank you,
Mr.
sir. Thank you.
Anil Kumar Bhatnagar.
Oh, thank you very much.
Congratulations, Mr. Anil
Congratulations,
Kumar
sir.
Bhatnagar, sir. You have
Thank
given a
you.
lot of recent what we can say recent concepts about misconception also in the current variant of COVID-19 pandemic. So congratulations, Mr. Anil Kumar Bhatnagar, sir. for
Thank you
getting
very much,
this
sir.
award. Congratulations. Thank you. Next.
Thank you, sir. Congratulations to all the participants and all the best for future. The end of a story is the new beginning for many others. On this note, let's conclude a remarkable and knowledgeable valedictory session. On behalf of Faculty of Pharmacy, Oriental University, Indore, I sincerely thank all the dignitaries for being with us in this event and enlightening us with their enthusiastic remarks. Thank you, everyone. Have a wonderful day.
At last, I would like to say one thing. Again, my team is my proud. All of you have done a great job. Really, cheer up for all the team members, all the committees, when you host scientific invitation and media committee. So all the committee members done a great job, and really we are doing good to promote the research by organizing this kind of international conference, international faculty development program. So I congratulate all the faculty members of the Orient University Indore Faculty of Pharmacy and all the participants, those who have given their time to this conference as active participation. So definitely looking for more conferences, more workshops with the Faculty of Pharmacy, Orient University Indore, and accepting your full participation in future also. Thank you so much. And once again, thanks to all the international keynote speakers for sharing their valuable insights on the World Congress